Hello children, how are you all? I'm sure you're all doing well. Before we begin the class for today, let me give you a bit of introduction about this app. So this app is NEET Biology at Locus. This application, children, gives you coaching, especially on biology, that is on NEET Biology. So if you are a NEET aspirant and if you want to prepare effectively for NEET this year, please download this app. Now, if you want to know more about the app, I have provided a link in the description box. Children, you can click onto it and we will get in touch with you. Please watch my video till the end, and if you like it, please give a thumbs up, subscribe to my channel and hit the bell icon. So without wasting any time, children, let's get into the class for today. And the topic which I'm going to teach for you all, you can look onto the screen here. It's taken from CBSE 11th biology topic, which is a part of NEET syllabus. And I've taken it from Unit 1, which is Diversity in the Living World, and Chapter 4, that is Animal Kingdom. Now, I'm not going to teach you the entire chapter. I've taken a topic from Animal Kingdom. So let's get on to the topic now. And let me also tell you that this video is going to be in two parts, part 1 and part 2. And I want you to watch both the videos because at the end of part 2, I am going to share some questions which have been asked, in from NEET. So you would have an idea as to how are the question types and what all questions have been asked from previous NEET. Then, children, I'm also going to share the weightage for the topic. So don't miss it. Watch both the parts till the end. Now, what am I going to teach today? So I am going to teach about one phylum. I picked up one phylum for today. So before that, let me give you a bit of introduction about the animal kingdom. Now, if you know children, animal kingdom, we have two categories of animals. Now, as animals without vertebral column, we call them as invertebrates, and uh, these are the examples of invertebrates. Then we have animals which possess vertebral column. And what do we call them? We call them vertebrates. So this is very simple. You all know it. These are some examples I've taken from the vertebrate category. Now, as I said, I'm going to talk about one phylum. So before that, let me give you about the 10 major invertebrate phylum. So under invertebrate phyla, you have 10 major phyla, and these are all the 10. If you look into it, the first one comes Porifera, second is Nidaria or Coelenterata, third one is Ctenophora, the fourth phylum is Platyhelminthes, fifth one is Nematoda, sixth is Annelida, seventh is Arthropoda, eighth is Mollusca, ninth is Echinodermata, and tenth is Hemichordata. Now, are we going to learn all this? No, we are just going to concentrate on this one phylum, and that is Porifera. Let me tell you, students, this is a very interesting phylum. I suppose all phylums are. And we have, one of the most beautiful animals under this phylum. Now, since you are preparing for NEET, it's not like you should be very serious. Of course, you should be serious about NEET, but also children try to appreciate all the animals around us and the nature around us. Now, if you look at these animals, they are so beautiful. Now, I have put some pictures of these animals. Look at them. They look so beautiful. They all come in different colors. And where do they live? They live in water. So I would consider them as these beautiful animals in water or aquatic animals. And let me tell you, there are about 5,000 species of poriferans. 5,000, just imagine 5,000 varieties of only poriferans we find in water. Now, if you see, look onto the screen, children, there's one poriferan and it's called Euplectella. Euplectella is commonly called as the glass sponge. Why? Because it looks like that, so that's why they call it the glass sponge. And if you see, Euplectella is given as a gift, as a wedding gift in Japan. Now, why do they give it as a gift in Japan? You would come to know the answer at the end of the video, that is end of part 2 video. So hold on to it and know the reason why people give these, you know as a wedding gift. So we are going to learn about phylum Porifera, which comes under subkingdom Parazoa. So before we get into the details of it, children, let's learn about the animal kingdom. Now, under animal kingdom, we have two subkingdoms. One is Parazoa and the other is Eumetazoa. Animals are also called as metazoans, which are multicellular organisms. Under metazoa, you would have Parazoa as one subkingdom, and the other subkingdom is Eumetazoa. Now, why are poriferans kept under Parazoa? The reason is parazoans are a separate, organisms, or you would say animals, though they are multicellular, but they do not show tissue organization. That means what? These animals do not show any tissues. Like we would consider these animals as the most simplest animals on earth. Just imagine they don't have tissues, they don't have organs, they don't have organ systems, they just have a group of cells performing different functions, but yet they are animals. So that's why scientists have kept them under a separate subkingdom and they are called as parazoa or parazoans. Now, on the other subkingdom, you have eumetazoans, where you have animals having tissue level of organization or they show tissues, organs, and organ systems. Now, parazoans or poriferans, they are called as an evolutionary blind offshoot. Now, why do you call them as evolutionary blind offshoot? Is since these, sponges, their ancestors do not give rise to any major groups. Now, if you talk about other ancestors like our ancestors or other animals' ancestors, they give rise to two or the other major group. For example, you take bacteria and Arachibacteria or Archaebacteria. They have a common ancestor and they are considered as anaerobes. So anaerobes are the ancestors for these two major groups, which are bacteria and Archaebacteria. But when you talk about poriferans, poriferans ancestors do not give rise to any other major group. Now, who are the ancestors of poriferans? They are these unicellular organisms called as the choanoflagellates. So, these are single cells having a single flagellum. So, scientists say that choanoflagellates are the ancestors for poriferans. Now let's learn about the scientists. Scientists have greatest contribution towards discovering these beautiful organism, any organism. Now for Porifera, you can see Robert Grant established this phylum Porifera. So he was the one who did so much of study and so much of research on these beautiful animals, and then he established this phylum. And this is the person. Right. Now, what is the meaning of porifera? You can see porous means pore because their body, if you look at their body of sponges, they are all holes or openings surrounding their body. So that is why porous means pore, ferrous means pore bearing organisms. Now, there's some significance why their body has, these pores or holes around. They are commonly called as sponges. So instead of calling them porifera, you can call them as sponges. Now there's another scientist, and his name is John Ellis. John Ellis is the first person to recognize sponges as animals. Children, before this scientist, all others they believe that poriferans or sponges can be plants because one point about sponges are they don't move, they are non-motile. So since they are attached, they are non-motile, they were considering sponges as plants. But this is the first person who observed, who did a detailed study and research, and then he said, no, no, sponges are not plants, but they are animals. And then they are generally marine. You would not see sponge on the land, but you would see them in water, and that too in sea and in oceans. Study of sponges is called parazoology. So since they are kept under the separate subkingdom, the study of sponges is called as parazoology. This is how a sponge looks like, and these are some examples. Okay. Now sponges are multicellular, as I told you, and they are sessile. What do you mean by sessile? They do not move. They are immobile. So they cannot move from one place to another. They are always attached to the substratum under the seabed or ocean bed. Now the next point about sponges are some animals are radially symmetrical and some are asymmetrical. Now, let me tell you, children, most of the poriferans are asymmetrical, some show radial symmetry. Now, the point is, what is this symmetry all about? So, I'm going to talk about this symmetry, children, for those who don't know. Now, symmetry is, it talks about how the internal organs of plants and animals are arranged. Remember that there is a particular pattern which follows where certain organs are arranged in a certain pattern, which we call it as symmetry. Now, symmetry is divided into different types, like one is the radial, the other is asymmetry, and there is another one, bilateral symmetry. Although I'm not going to talk about symmetry right in this topic, but the next video that I'm going to make, children, is all about symmetry. I want you to watch it and learn about symmetry. Now look at these two pictures. You can look onto the screen. The left side picture, these are poriferans or sponges which show radial symmetry, and the right side ones show asymmetry. So for all those who don't know, let me give you a little bit of introduction about this symmetry. Let's go back. Let me talk about radial symmetry. So if you look at the screen, children, radial symmetry is also called as monopolar monoaxial heteropolar symmetry. Now, what is the meaning of monoaxial heteropolar symmetry? The meaning is, I want you to follow this my cursor, children. Now look at this picture which is showing radial symmetry. Now, radial symmetry is exhibited by organisms who are usually sessile, that means who do not move, and whose body is basically cylindrical. So, if you look at sponge, some of the sponges have this cylindrical shape. Okay. Now, when you cut an animal, and when I say cut an animal, you always consider symmetry in the embryonic stages. You don't consider in its adult stage. So we take the embryo of this sponge, and then from the center point. Now this center point is called as the central axis. There are many names to central axis. You can call it as principal axis. You can also call it as oroboral axis, right? There are many names to it. So what is the central axis? It's an imaginary point or the central point at the body of the organism, and from the central point, if you try to cut the organism from any plane, like you can see, you can, if you're trying to cut it, just follow my cursor in this way or in this way or in this way. When you try and cut it, and when you observe the two halves, you would see mirror images forming or identical pieces. So such kind of symmetry is called as radial symmetry. So if an animal is exhibiting radial symmetry, you can cut the embryo of that animal from any plane and you would get mirror images. Now, how are we getting mirror images and why are we getting mirror images like that? You would all learn in my next video, so please watch it. So for now, I want you to remember some of the sponges, they show radial symmetry and some of them do not show symmetry at all. Like which ones? If you look at this picture, asymmetrical, like sponges which are shapeless, they show asymmetry. So the animals which lack symmetry are called asymmetrical animals. Now, if you look at this here, children, the highlighted point here in red, it says axis is single, poles are different. Now, what is this axis all about? The central axis, and poles is from any direction. If you're trying to cut the embryo of the animal, you would get this symmetry. Now we go back to the point. The next point, if you look at it, these organisms show cellular level of organization. Now, what do you mean by cellular level of organization? As I said, these animals are multicellular. They show cells but no tissue formation. Now, if you ask me what type of cells do these animals have, they have these cells called as choanocytes, porocytes, pinacocytes, tesocytes, amoebocytes. Although I'm going to talk about some of these cells in the coming slides, but just remember, children, they are these group of cells which are doing different functions. I'll talk about amoebocytes. Amoebocytes are totipotent cells. Now, what are totipotent cells? These are stem cells. which give rise to other cell types. These are undifferentiated cells and they have the capacity to give rise to other cells. Like these amoebocytes give rise to another kind of cells called as cells. Now, what are the function or what is the function of these cells is they store food. Like that, there are different group of cells in poriferans and they have different functions, which I'm going to talk in the coming slides now. So cellular level means they show only group of cells, but no tissue organization. Then if you look at their body, children, their body is composed of two layers, or their body wall is made up of two layers. The outermost wall is called as pinacoderm and the innermost wall is called as choanoderms or choanoderm. So they are showing two layers in the body wall. Since we are done with it, I'm skipping this slide. And look at this picture, children. This is how a typical sponge looks like, and these are the parts, right? Now, if you look at the amoebocytes I was talking about, amoebocytes are the ones which are, they are undifferentiated cells and they give rise to many other cells. One of the cell is thesocytes, as I told you. Now, if you look at the body of the sponge, the outer layer of the sponge, you know, it is called as pinacoderm. Now, why pinacoderm? Because it is containing certain kind of special cells called as the pinacocytes. Look at the picture here, children. These are pinacocytes which are lining the outer wall. And in between these pinacocytes, you would see another special type of cells called as the porocytes. Now, what will these porocytes help in? Remember, porocytes are the cells, through which water would enter. Since these are marine organisms and they are attached to the substratum, they cannot move. So, water enters through these porocytes. And the opening of these porocyte is what we call it as ostia. So ostia are these small pores present on the outer layer of these sponges. So water enters through ostia and then water exits through a large opening called as osculum. And every day water enters through ostia and exits through osculum. Now there's one interesting point that I want to say here, children. It seems Porifera in 24 hours, it pumps about 24,000 liters of water. 24,000 liters of water is pumped by 1 kg sponge in 24 hours. So you can just imagine so many liters of water is pumped. And while it is pumped, what is happening is there are many other functions which are carried out, which I'm going to talk in a while. Now, I want you all to observe there is a central large cavity here, which is called as spongocele. Now, cele means what? It's a cavity. So, the cavity of sponges is called as spongocele. Now, there's another layer here which is called as mesohyl layer. What is this mesohyl layer? Between these two layers, there's an undifferentiated layer called as the mesohyl layer. Now, it is in these layers you will find these amoebocytes and the other different types of cells like the thesocytes. Okay. Now let's get into another important point. See, sponges, as I told you, they have this large cavity inside and which is called as spongocele. You can also call it as a canal system, a water transport system, or a paragastric cavity. You can use any word to describe it. Now, what is the main function of this ostia, spongocele, and osculum? I'm going to talk about it. Now, look at the second point. Water enters through ostia, then moves into spongocele, and then exits out through osculum. Now, children, I told you about the external layer that is the pinacoderm. The internal layer is called as the choanoderm. And remember, choanoderm is lined by very special cells that you would find only in sponges and they are called as the choanocytes. So you can see here choanocytes are also called as collar cells which line the spongocele and the canals. They are flagellated cells. Right. Now, what is the function of this spongiocele? So, as I told you, this spongiocele is lined by choanocytes. So, whenever water enters through ostia, if there is any food particles in the water, the particles is captured by these choanocytes, right? They would take it and they would digest. What they would digest? They would digest the food material. So, choanocytes are actually helping in digestion. In the same way, when the water is entering ostia, many other functions are also happening, like respiration. That means exchange of gases would happen since they are animals. They would take in oxygen and give out carbon dioxide. How is the water exiting? Out through osculum. There is circulation happening, circulation of many things like the food is getting circulated. There's excretion happening, nutrition and reproduction. Even reproduction happens through, when the water is entering and exiting. Now, the nutrition is holozoic. Now, what do you mean by nutrition is holozoic, children? Always remember, holozoic nutrition means uh, it's a kind of nutrition where we ingest the food, that means we take in the food through the mouth, and once the food enters the different parts of the digestive system, the food gets digested. Once it gets digested, the food is absorbed and then assimilation takes place. So, digestion, assimilation, right, happens inside the digestive system. And then there is the final thing is excretion. So, undigested matter comes out. So, this kind of nutrition you call it as holozoic, where we are taking the food inside and the food is getting digested inside the body. That is holozoic. Now, when it comes to sponges, sponges don't have a digestive system. How does it happen? See, digestion is intracellular, the second point says. That means what? The cell, like the choanocytes, they would take in the food, they would digest the food within themselves. So you call it as holozoid. The digestion is happening inside the organism and the food is getting into the cell, that is intracellular. That is what is holozoic and intracellular kind of digestion. Thank you.